Hello everyone, my name is Tizian Schulz and today I'm going to talk about sequence-based pangenomic core detection. This is a joint work together with my colleague Roland Wittler and my supervisor Jan Stoye from Bielefeld University. If we compare different individual genomes that are closely related, for example, because they come from the same species, we may find that they actually still vary a lot. And this gives rise to the topic of Pangenomics, which was first introduced by Thaliana et al. In 2005. They introduced two new notions. First, the notion of the pangenome, which is the set of all genes from all genomes of some species, and the core genome, which is then the subset of genes that occur in all these genomes. The pangenomic core has a lot of applications. For example, it can be used to study genetic diversity. It can be used in drug development and for vaccine design, and it also has some applications in crop plant breeding. However, it also comes with some drawbacks. First, it requires an expensive pre-processing. If we have sequenced our genomes, we need to assemble them first, and then we need to find genes within these assemblies. And what is even more difficult is then to map the genomes so that we can find which genes are actually shared between the genomes. Also, we cannot find any core features which are below or above the gene level. And this might be okay if we have a prokaryotic species where actually most of the sequence is coding. But if we consider eukaryotic genomes, then this might mean that we miss a lot. This is why we went to a sequence-based pangenomics approach, where a genome is no longer a set of genes, but a set of strings, which can be short strings like sequencing reads coming directly from a sequencing machine, or completely assembled chromosomes. And this has some advantages. First of all, we don't need any gene annotation. We also don't need to map the genes. But still, we do consider genes implicitly because, of course, our sequences will still contain the genes. So it's the approach is kind of a generalization from the gene-based approach. And now, finally, it also makes sense to consider eukaryotes in this setting. A sequence-based pangenome can be represented by a sequence graph, for example, by a color-to-brown graph. In a color-to-brown graph, each vertex is a substring of length k, which we also call k-mer. And the vertices of the graph come with a set of colors. And the colors tell us from which of the initial input sequences the k-mer came from. Then there is an edge connecting two vertices in this graph if the vertices share a k-1 overlap in their sequence. Using such a graph to represent a sequence-based pangenome again has some advantages. First of all, we don't need any genome assembly. We can use sequencing reads from our sequencing machine and put them into the graph, and the assembly somewhat comes for free. Because every k-mer is only stored once in the graph, this also reduces the redundancy in our data set a lot, and it leads to a small memory footprint. Now the question is, if we have such a graph, how can we find a sequence-based core inside it? And the most straightforward thing is what you can do is that you take such a graph that represents a pangenome, and then you choose an integer which is between 1 and n, where n is the number of genomes in your pangenome. And this integer you call a quorum. And then you say every k-mer that occurs in at least q genomes, q individual genomes, belongs to the core. In this case, we call this set of k-mers the core k-mers. And if we look on the example down here, where k is 3 and our quorum is 2, then we find 3 core k-mers in this graph, which have a color set of size 2 and larger. However, this definition also has a problem, and this I try to visualize by this multiple sequence alignment down here. So we see a multiple sequence alignment of 5 homologous genes. Doesn't matter so much where they actually come from, but they are homologous, and we see that they actually share a lot of their sequence. Most of the sequence is conserved within these genes. However, they have some positions where there is some variations. And this is a problem for our upper definition, because these parts that have some variation, they won't be part of our core. So we won't be able to find the complete genes inside our core, even though they should be in there. They are homologous. They should be in part of our core. So what can we do? We decided to use a second set of k-mers to deal with this problem. And for this second set of k-mers, which we call bridging k-mers, we introduce a second integer, which we call delta. And then we say every k-mer in our graph is called a bridging k-mer if it lies on a path that connects 2 core k-mers, and the path is no longer than delta plus 1. In our example down here, this is true for 6 k-mers, which lie on paths that connect k-mer GCA and TTA in the graph. And the paths in this case are of length 4, which is delta plus 1 in the example. For the upper k-mers here, they also lie on a path that connect 2 core k-mers, GGC and TTA, but the path is too long. Now we say both sets together, the set of core k-mers and the set of bridging k-mers, we call our core genome. And now we also can deal with variation inside our sequences. The problem that we now need to solve is if we have a pangenome that is represented as a color to brown graph, and we have 2 integers, a quorum and a delta, to find the 2 sets of k-mers, the set of core k-mers and the set of bridging k-mers. How can we do this? Well, to find the set of core k-mers is straightforward. We can just iterate over the graph, and for every k-mer, check the size of its color set. If it's at least of size of the quorum, this k-mer is a core k-mer and it belongs to our core. The detection of bridging k-mers, however, is more difficult. What we could do is we could try to enumerate all the paths in our graph that connect 2 core k-mers and then check their lengths. And if the length is short enough, then we could mark all the k-mers on that path as bridging. However, this is something that we don't want to do. We definitely don't want to enumerate all the paths in our graph, because there might be an exponential number of them. Luckily, we don't need to do this. If we use a special kind of graph traversal in which we annotate every vertex with the shortest distances to the next core k-mers, we can avoid the enumeration of all the paths. So how does this traversal look in detail? Well, we go back to our example from before. And the first thing that we do is we make an abstractions step. We forget about the explicit labels of the vertices, and we replace them just by some unique identifiers in order to make it easier to understand the traversal. In order to understand the traversal, we don't need the actual labels of the vertices. And also what we do is we add a Q data structure here, which is initially empty. So the first thing that we do is we try to find all the core k-mers. And this, as I said before, can be done with a simple iteration over the graph. So we start iterating over the graph, assuming here we start with A. We check the color set of A. And the color set in this case is 3, which is larger than our quorum. So A is a core k-mer. And now we can annotate A with the shortest distance to the next core k-mer, which, because A is a core k-mer itself, is 0 by definition. So we annotate A with 2 integers. One integer represents the distance to the next core k-mer if we use outgoing edges, and the other one represents the distance to the next core k-mers if we use incoming edges, which in this case we do not consider for this example. Now, after we have annotated A is added to the queue, and we continue our graph traversal. The next vertex in our graph is B. B has only one color in its color set, and this is smaller than our quorum, which means that B is not a core k-mer. At this point, we cannot annotate B, so B is not added to our queue, and we go on. The next vertex is C. C has 2 colors in its color set, which means, again, that C is a core k-mer. We can annotate C, and then C is added to our queue. In the end, after the initial traversal of the graph, we have found all core k-mers, which in this case are 3. And the core k-mers are annotated, and because they were annotated, they are also inside our queue now. Next, we want to annotate all the other vertices in the graph, which might be potential bridging k-mers. So we start with the first element in our queue, which is A, and we go to A in the graph. Now for A, we check all its neighbors in order to annotate them. So the first neighbor of A that we do consider is B. And because B is not annotated yet, we can annotate B with the distance to the closest core k-mer if we use incoming edges, which is the shortest distance to the next core k-mer using incoming edges of its predecessor plus 1. So in this case, 0 plus 1 equals 1. Because we have now annotated B, B is also added to our queue. And we continue with the next neighbor of A, which is C. C is already annotated. And because of the special nature of our traversal, we know that if a vertex is annotated, it is already annotated with the closest, the minimal distance to the next core k-mer. So we don't need to update any annotation in our graph. Since C is already annotated, we don't need to annotate it again, and C is also not added to our queue. After we have considered all the neighbors of A, we go back to the next element in our queue, which is C in this case. So we go to C in our graph, and we check out its neighbors. One neighbor is E. E can be annotated in the same way as we annotated B before. And because E is annotated, E is also added to the queue. The same goes for F. And then one more neighbor of C is A. A is already annotated, so A does not need to be annotated again, and so it does not need to be added to the queue. The next element in our queue is M. M has no successors that we want to consider here, but it has 3 predecessors. So we can annotate the predecessors with the distance to the closest core k-mer considering outgoing edges. And we do this with all 3. And because we've annotated them, we add them to the queue. In the end of our traversal, our graph will look like this. So we have annotated all the vertices in the graph. Not all vertices are actually annotated with 2 annotations, as you can see. This is because at some time we can terminate a traversal before we have annotated all the vertices. For example, here, if we would want to annotate B with the distance to the next core k-mer using outgoing edges, we would have to come from D. But here we see that the annotation of D is already 3. And if we would annotate B, we would have to annotate with 4. But B is not a core k-mer and our delta is 3. So we know that B can never be part of our core. It cannot be a bridging k-mer. So we don't need to annotate it. We don't need to add it to the queue again. And we don't need to consider its neighbors again. The last step of our algorithm now is the output of the core. We have all information we need already stored in our graph. So the only thing that we need to do now is to iterate over our graph once again and use the annotations we put there. So let's assume we start with A again. What we do is we add up the two annotations of A and compare it to delta. And if it is not larger than delta plus 1, this means that the vertex is part of our core. In this case, 0 plus 0 is smaller than delta plus 1, so A is part of our core. The next vertex is B. B has only one annotation, which means that B cannot be part of our core. Then we consider C. C again has two annotations, and the sum of the annotations is smaller or equal to delta plus 1, so C is part of our core. In the end, we have found the complete core that we wanted to find. And I'd like to stress once more that this was possible without the enumeration of all the paths in the graph. The only thing that we did was to traverse the graph several times. One time to find the core k-mers, then we used the queue, and in the end we traversed the graph once more to add up the annotations for each vertex to find the core k-mers and the bridging k-mers. So the first and the last traversal are possible in or are linear in the number of vertices in the graph. And the step in the middle that involves the queue is also linear in the number of vertices in total. Why is this? Well, a vertex is only added to the queue if it was annotated before. And every vertex is only annotated at most twice. So in the end, we end up with a linear runtime for the whole algorithm. With some modifications, we can use the same algorithm also with a compacted color-to-brown graph instead of a color-to-brown graph. First of all, what is the difference between both graphs? So in a compacted color-to-brown graph, we merge all the vertices that lie on a non-branching path in the color-to-brown graph. So we end up with a graph that has much fewer nodes. And with some modifications of the algorithm, we can, as I said, use the same algorithm also for this kind of graph. However, the asymptotic runtime increases for the compacted color-to-brown graphs. This is because color sets are now no longer assigned to vertices in the graph, but to the k-mers. And there can be many k-mers in the label of a vertex. However, in practice, the compacted color-to-brown graph has much fewer nodes than the color-to-brown graph, which means that it is, even though it has a larger asymptotic runtime, is faster in practice. And this is why we implemented the algorithm for the compacted color-to-brown graph, and our implementation is called Cora. We tested our implementation in comparison to other related approaches. First, we used an approach that is actually the classical gene-based approach. So Panaroo calculates a pangenomic core that is gene-based. And then we used also a tool which is able to calculate a sequence-based pangenomic core. Sibelia Z is actually not a tool that was developed for doing this. Actually, it was developed to find synteny blocks within multiple genomes. But if we add a quite straightforward post-processing step, we can also use it to predict sequence-based pangenomic cores. And we did so because, as far as we know, there is no other approach except from our own approach that is actually able to calculate a sequence-based pangenomic core yet. Then there were also two other approaches that we wanted to compare against, but due to different reasons, this was not possible, unfortunately. The initial data set that we used for the evaluation consisted out of 4 prokaryotic pangenomes of different sizes and different levels of sequence diversity. As parameters for our algorithm, we used a quorum of 95% with respect to the size of the pangenomes and a delta of 60. And then we added parameter t, which we used to map results from the gene-based approach to results from the sequence-based approaches. We said a gene was found by the sequence-based approaches if its sequence appeared by at least 50% inside the sequences that were predicted by the sequence-based approaches. Our experiments we performed in our compute cluster in Bielefeld and also partly on the Denby cloud. And here is the result. So we see for each of the 4 data sets a comparison, a pairwise comparison for all combinations of tools that we considered. And the blue surface represents the number of genes that were shared within the cores of both tools, whereas the red and the blue surface is the number of genes that were unique for within the result of the respective tool. So for the first 2 data sets, we see that all the tools agree quite well. Most of the genes they predict are actually shared. For the larger one, or the larger 2 data sets, we see that Sibelia Z had some problems. Its core was much smaller for these larger data sets, and this is why we see only a very small overlap of the results and many more genes which are predicted by Cora and Panaroo in this case. For the largest data set of monocytogenes, Sibelia Z was reporting a core that was so small that there were no genes inside this core at all. So we omitted to show the bars here. If we look on runtime and memory usage, we see that Sibelia Z actually does a good job. Well, in terms of runtime, it does a good job. Here, Cora is much larger. However, one has to say that Cora is using a much smaller k-mer size than, for example, Sibelia Z in this case, in order to have a better sensitivity. So if we would also use a larger k-mer size like Sibelia Z here, probably we would be also much faster. But since the absolute runtimes are still small, we could afford to go for a lower k-mer size and get a better sensitivity here. On the other hand, if we look on the memory usage, we see that Cora actually is only taking very few memory compared to the other 2 approaches. We also studied the influence of our delta parameter on first the result and also the runtime and the memory usage. For this, we used the largest prokaryotic data set and we calculated its score for increasing values of delta. What we see is that the larger we choose delta, the larger our core gets until at some point a saturation is reached. And we also see, if we compare this to the runtime and the memory usage, that both actually correlate quite well to the size of the output. So this is one thing that we can hold, that the runtime and the memory usage for increasing values of delta is mostly influenced by the output size. We also did some experiments for eukaryotic data sets, And here, for example, we used a data set consisting of 18 accessions of Arabidopsis thaliana. And we used Cora and Sibelia Z to calculate cores for this data set. So if we compare both cores, we see that on the level of k-mers, they agree quite well to each other. And also on the level of genes, we see that most of the genes are shared, whereas Cora was able to predict some, to find some more genes inside its core. For this data set, we also see that Cora this time, even though we still use a smaller k-mer size, is faster than the Sibelia Z. And again, it uses less memory. Here we also had a look on non-gene core elements, and we could find that part of our core were most of all non-coding RNAs, some pseudotranscripts, and also transposable elements. This was reported by both tools. We also wanted to study the influence of assembled data versus raw read data on the quality of the core that we predict. And luckily, for 17 out of the 18 accessions of our Arabidopsis thaliana pangenome, we also had the read data that was used to generate the assemblies. So we were able to build two different pangenome graphs here. For one graph, we used only the assemblies, and for the other, we only used the read data sets. And what you can see here is the results for both graphs. So first of all, we see that the pangenome graph for the read dataset is much larger than for the assembly dataset. And also, it took much longer to calculate a core for this read graph. Also, we used more memory. But then, if we look on the result, we see that the result is actually quite similar, no matter if we use assemblies or reads. So most of the k-mers inside both cores are shared, and there's only few k-mers more inside the read graph compared to the assembly graph. On the level of genes, also most of the genes are shared. If we look on the genes, we also had a special look on a certain set of genes, which are called Busco genes. Busco genes are genes from which is known that they have an ortholog in all species of some taxonomic unit. In this case, we use the order of Brassicalis. So this means that actually all Busco genes should appear in the core of our pangenome, because all accessions of Arabidopsis thaliana belong to this taxonomic classification. However, 15 Busco genes were missing in the core of the assembly graph, and only 7 were missing in the read graph. Okay, we are at the end of the talk. I'd like to summarize what I said before. So first of all, we saw that sequence-based pangenomic core detection is possible without the enumeration of all paths in the graph, and that means it is possible in linear time. The usage of bridging k-mers allows us to handle varying degrees of sequence variability, which is a nice thing. The use of a graph in turn reduces our memory requirements. I guess this is something that we could see in all the experiments that I've showed you. And also it enables us to use read data. And read data, if we use this for our pangenome, it increases the computational demands. But on the other hand, on the side of the result quality, we see that actually it produces comparable results. So it is definitely not necessary to assemble all the genomes if you want to predict a sequence-based pangenomic core. That's it. Thank you for your attention.